Right. So when we talk about a gene being expressed, so gene expression basically consists of, we have a gene that is going to be converted into mRNA, and which is going to be converted into tRNA, that is going to eventually form the protein, and that protein is basically the expression of the gene, as we've talked before, right? So this process is basically known as the transcription of the gene, and this process is basically known as the translation of the gene. Now, if you've wondered, or if you've ever wondered, that there are certain proteins that are only made in certain parts of the body. For instance, HCl is only made in the stomach. It is not made in the eyes. There is no HCl within the eyes, whereas the eyes have vitreous humor, which is not made in the stomach. Now, why is this so? We have studied all our lives long that each cell in our body have the same set of DNA. Then why is it so that the stomach cell produces HCl but the eye cell does not produce the HCl, even though it has the gene for the production of HCl and it has the same DNA as that of the stomach cell? The reason for this is actually the control of gene expression. The stomach has that control that it would only express those genes, it would only switch on those genes that produce HCl, and it is going to switch off those genes that produce the vitreous humor. Similarly, the eye would switch on those genes that produce the vitreous humor but switch off those genes that produce the HCl. That does not mean that the cells in the eye do not have the genes for the production of HCl. It simply means that the genes for the production of HCl are switched off in the eye cells, whereas they are switched on in the stomach cells. Now, we need to talk about how does this switching on and switching off go about. In order to understand that, first you need to understand a basic thing, that is this, that if we talk about the replication of DNA, if this is DNA polymerase and we need to replicate a strand of DNA, how is it going to replicate the strand? From our prior knowledge, we know that DNA polymerase need a primer, which is actually a guidance for it where it needs to attach and from where it needs to start replicating the DNA strand. So DNA polymerase is going to come and join over here on the primer, and then it is going to carry on its replication in the forward direction. So the primer plays a role in order for the DNA polymerase to guide it where it needs to bind to the DNA strand and from where it needs to start its replication. Similarly, when talking about the production of mRNA from the DNA strand, we need RNA polymerase, because mRNA is a RNA and polymerase means that a RNA chain that is being made, right? Poly is from polymer, ase is for the enzyme, and RNA polymer is being formed. So many mRNA are being formed in the form of a chain, right? So if this is the DNA strand, how is the RNA polymerase going to bind to it? Because it would not know where it needs to bind. So if these are the genes that needs to be transcribed, that needs to be transcripted into mRNA, we have two main things behind this gene. We have a promoter region and we have an operator region, right? Now, the function of the promoter region is actually to guide the RNA polymerase like the primer so that it can come and bind to it and then start its polymerization and then start its function. But over here the regulation is dependent upon the operator. How? Let me erase some things from here. Why isn't this working? What is happening? Just a second. Isn't this erasing at all, man? Okay, just a second. I have a technical difficulty.